neurons. All right. If the lecture goes blank, warn me this time, okay? Don't let me speak without having the lecture on like yesterday. Cool. We are almost all there, actually, I think, because you're 44. The people in the lab are going to check your presence. So, you know, we're going to know very soon who is there and who is not. But before we go on, do you have any question or comments or anything that stems from yesterday? Could you all download the software that we're going to use today, Neuronify? And you open and it works. Perfect. Okay. So I guess you're all set for today. Right. So I can start sharing my screen for the lecture. So one warning, I drank way too much coffee this morning. So if I speak fast, please let me know. Like really, let me know. I might need it. Okay. First of all, a couple of announcements. So first is remember to document your experiments in the lab notebook. For example, if I would ask you right now in the Hodgkin-Huxley model, which was the intensity of the stimulus that did not elicit an action potential, you should be able to go back to your notes and tell me the exact number, because that's what I want to see on Sunday when you submit it. The assignment of today has been slightly updated because I think you really are good to work on your own. So please download it again from Adam. And also in Adam, in the folder week 4 papers, you will find which paper has been assigned to you for the discussion of Thursday. So you can already start reading it. There is a time slot allocated to the paper reading on Thursday, but it's definitely not going to be enough to only read it at that moment. So go there, try to read it before and refresh it on Thursday. All right. So today we're going to talk about and understand how neurons talk to each other and how we can listen to all the chatter that goes on in our brain. We're going to talk about biological neural networks and the way in which the way we connect neurons to each other not only defines who speaks to whom, but it also defines the coherence of the sentences that are said between neurons, the timing of the sentences that are said between neurons. And in last instance is really what the lump of fat tissue that we have in our skull does, right? Decide which neuron talk when. So a little bit of refresher from yesterday. What did we learn yesterday? Yesterday we learned, and by now you are expert of action potentials, that this is the way in which neurons talk to each other. These are electrical signals that are generated by maintaining and dissipating an electrochemical gradient of ions between the inside and the outside of the cell. Not all the cells work the same way and decide to fire an action potential in the same way. The ones that are at the periphery of our nervous system are able to pick up signals from the physical world that usually are change in energy of different domains, can be a sound wave, can be light, can be heat, can be a smell, an odorant, and are able to convert this information into an electrical signal. When we move within the brain instead, in the CNS, the rule for transmitting information are slightly different in the sense that one neuron receives thousands of information through its spines. And depending on the local state of the dendrites, its history, and the strength of the synapse, then we can get different, every neuron has different weights. Which means that different probability to cross the threshold for an action potential. We are going to talk about this today. So if this concept is not completely clear, it should be because you've already seen it. But if it's not clear, we're going to revisit it in a few hours. Also, the signal that the message can transmit can be either positive or negative, can be excitatory or inhibitory neurons. The message from the synapses then are integrated, processed, and if it crosses the threshold for finding an action potential at the soma action segment, then the message is transmitted. Excitation and inhibition interact with each other. You have spatial summation, you have temporal summation, and you might have cancellation of signal given by a coincident excitatory input that arrives with an inhibitory input. And you will see today with your biological neural networks how this is actually really important in deciding if the neuron will fire or not. But the reality is that yesterday we have modeled one neuron, we have modeled one area of the axon into an action potential, but this is not the way in which neurons really work. If we look into the brain, the thing that we see, it's even this is an oversimplification. We see a lot of different neurons that start to talk and connect to each other. So neurons are not isolated entities, but they connect to thousands of other neurons to create neural networks. The simplest neuron consists of two synaptically connected neurons. That's already considered to be a very simple network, the synaptic network. And this is something, for example, that you experience every time you have a reflex. You know, the stimulus that goes from your sensory neuron in the shortest path, it can cross just one synapse and elicit a response. But to create complex cognitive function, that is the things that we are interested in this course and in our lab, you have definitely more than two neurons and receive input and send out input to more than one partners. Especially things get complicated when you have excitatory and inhibitory neurons that talk to each other, because then in that case, you need to understand really like who talks to whom and at what time. So if we look at the brain from a single neuron perspective, and we look at an area that is particularly close to the heart of the people that work together with me, CA3, you find that one single CA3 pyramidal cells is able to talk to 3,006, or better, receives inputs from 3,600-ish, you know, it's not so precise, but there is an estimation, cells from an area of the brain that is called the entorhinal cortex. It receives 12,000 synapses from other CA3 neurons, which are basically neurons that are located in an area of the hippocampus in our brain, which is called cornus ammonis area 3, that's where CA3, and then transmit information to a lot of other neurons. So in the end, one neuron of the area CA3 can listen to up to 15,000 other cells. How can we regulate the way in which neurons talk to each other in a way that makes them say coherent sentences? So how can we make sure that neurons speak to the right person and at the right moment? You can guess it, it's all in the connectivity, right? The way in which neurons connect to each other, make sure that the messages passed along specific routes in the brain, reach who needs to listen that specific message, and it's transmitted at the right time. If you look at the area CA3 here, it is embedded in a network in which it receives information from this area that I told you is the entorhinal cortex. You will hear tons about it on Thursday and tomorrow as well a little bit. It receives also information from the dentate gyrus that's presynaptic to it and is able to transmit information to area CA1. But in most cases, you have the area CA1, then unidirectionally moves this information again toward the entorhinal cortex, but does not goes back to CA3, although of course there is exception to every rule in the brain. This particular arrangement is not only useful to make sure that CA3 gets information from the dentate and transmitted to CA1, but also to make sure that CA3 neurons talk at the appropriate moment when the stimulus goes through the entorhinal cortex. And this is something that, again, we are going to explore in detail in the way in which it works both tomorrow and Wednesday. And Thursday, sorry. All right, so connectivity matters. The way in which you hook up neurons to each other really matters. So let's look at some of the principles that basically regulate the way in which the brain is wired up, right? First of all, I just want you to appreciate one thing. There are billions of neurons in our brain, and there are trillions of synaptic contacts. And all these neurons and synaptic contacts are created during development, and they're hooked up in a way that allows cognition to arise. And I always stop to think about this detail, and I'm always fascinated by it. So this is actually something, if you think about the brain as an amazing organ, it is. So you should really think about, be excited about that. Anyway, going back to our connectivity, let's start from the macro scale. So the global connectivity of the brain. As I said before, one big principle is that brain areas are connected to each other in a way that is able to create a stream of information from the outside world to the central nervous system, and from the central nervous system to the muscles, so that we can do the loop that we discussed about yesterday. You can perceive the word, you can attach meaning to this word, and you can decide what to do, and then elicit a behavioral response. In this case, we see the path, part of the path that goes from your eye to your visual cortex. Your eye with the retina and with the photoreceptors that we saw yesterday is able to pick up what's inside, what's in the world around you. it transmits this information through the optic nerve to the thalamus. From the thalamus, this information is transmitted to the cortical area that is called the primary visual cortex, or V1. In the cortex, this information is basically transmitted to different areas. And in the next slide, we'll see one of these principles. When this has made sense of what happens, then this information is transmitted to midbrain, and you're probably going to hear more with Sylvia next week about this. Then to the spinal cord, and from the spinal cord to the muscle. So in the end, you have a stream of information that goes from the outside world into your brain, and from into your brain through the spinal cord into your muscles. So you have the full loop, right? Obviously, this is a simplification. You have tons of shortcuts. You have tons of redundant loops. You have tons of feedbacks. But this is something that the general principle is that information goes in, information goes out. In the mesoscale, that means like all over the brain. As we saw before, there are connectivity loops that basically connect areas of the brain to each other. One of the examples was the thalamus connecting to cortex, connecting to the midbrain, right? But of course, there is also connectivity loops that are within the cortex, which are extremely important, from where information that comes from the senses moves from the primary area of your brain, for example, in this case, the primary visual cortex that we saw before, to areas of the brain in which information is not tuned to one specific sensory modality in a dominant way. It's not exclusively tuned to visual cortex, but it's predominantly dominated by that. But these areas that are called secondary areas, fourth and tertiary areas, association areas, and so on, become more and more multisensory as we go forward, and are more and more influenced by internal variables that can be experience, that can be the state of the animal, that can be the goal that the animal wants to reach at that moment, attention, arousal, you name it. This is something that you're going to see tomorrow. Okay, I lost the slide again, correct? Because I think Zoom crashed. Or are you seeing my slides? You do. All right. Because I get a message that Zoom
Can you, Flavio?
Yeah.
Can you switch one slide, because I think it's just stuck.
Yeah, I'm having strange stuff with my computer.
So are you clicking through slides now? Or?
No, because
okay.
my Chrome is behaving badly.
Now we're out.
Okay, no, yeah, you're lost me. 1 second, I need to restart Zoom. Does anybody has question about this part? Let's use it. Have you already taken a tour through the brain in different and other courses that you already know how information flows? So I assume this is a no because I don't hear anything. Do you see the screen now? Okay. All right, so now you see me, right? Okay, so let's go back where we were before. We are within the brain, we are in particular within cortex, and our information is moving from the primary areas to areas that are more and more multisensory, or that are more and more tuned to internal variables, or that are more and more able to create cognitive function. And this is something that we're gonna see tomorrow. But just to highlight you what the idea is that the messages come from your eyes to your thalamus to the primary visual area, then it moves to the secondary visual area, to the fourth, two areas that take different names, and you probably, if you're curious, you can go and look them up. V4, TEO, IT, SCP, prefrontal cortex, perirenal cortex, parahippocampal areas, entorhinal cortex, hippocampus. So the information flows to the so-called association areas in a progression that we'll see tomorrow why it has a specific function and meaning. Again, this is a simplification. I don't want you to get the idea that information moves only into one direction. You see that information flows in both directions. And this is going to be extremely important tomorrow and Friday, when we're going to talk about something a little bit more complicated. All right, but if we look at the mesoscale, we observe the movement of information related to vision. But you have to imagine that there is a parcellation of your cortex into areas that take care of different function. And within individual areas, there is a very precise type of connectivity where information is represented. For example, if you look at the primary motor cortex and the primary sensory cortex, you can see that there is sort of a topographical arrangement of the area of your body that are sensed in the sensory area or that are controlled in the motor area. It's not entirely the case. The point is that when we do microstimulation in specific area of the motor cortex, we elicit a response in a specific area, in a specific part of your brain. So again, I don't want you to think that you have a homunculus, how this is called in your brain, that does this in an exclusive way, but information is processed mostly in a way that is extremely precise and is represented in specific areas of the brain. If we go at the micro scale instead, that is locally within one individual area of the brain, that can be the hippocampus, it can be the primary visual cortex, it can be the cerebellum. The nervous system employs specific way in which neurons are connected to each other. And we call these specific ways in which neurons are connected to each other connectivity motifs. This is a way of approaching the brain that in system neuroscience we started to adopt in the last years, in which we are able to dissect the function of individual cell types and the contribution that individual cell types give to the way in which neurons communicate with each other. And in the way that we are going to do it, you will see today from a technical perspective and on Thursday as well. But the way in which neurons are connected to each other is extremely important because it creates and propagates, dominates the rule by which neurons integrate inputs at the level of the dendrites. So how do you should think about how this circuit motif works? You should think about it as having the ability to manipulate these coincidence arrival or inputs that can be excited or inhibitory that modulate the way in which a neuron is active. So if you have, for example, here a recurrent inhibition that we're going to go through this in detail, when this neuron excites this neuron, he is also able to excite an interneuron, which is shutting down a parallel pathway of information. So that you can decide that at the moment in which this neuron is active, only this path, the information propagates along the path that is identified by these two neurons here, while when this neuron takes over, information propagates toward this path, but not the other path. So this really creates channel by modulating the way in which dendrites are able, individual neurons are able to integrate the messages that they receive, and really shapes the way in which neurons are active, the temporal profile of activity that we discussed yesterday during the lecture about tetras. Because circuit motif in last instance decide when a neuron is active and whom it talks to because of the way in which it's connected, these are really fundamental computational units that are used to produce different activity patterns that are used to encode, process, and represent information within our brain. That's why during today, we're going to explore how this circuit connectivity motif work and what is the consequence of hooking up neurons in a specific way. Are we clear on this? Because this is going to be the whole day today. So if you have any doubt about this specific concept or why this is important, you should really ask me now. All right, so I don't hear any question. I'll just move forward. We are going to go now through all these motifs, look at them in details, and make, let's say, a comment of how they are employed in our brain and how they're actually used. You will see your exercise today, I'll already anticipate, will be to use Neuronify to build all of them and to observe how activity changes as a consequence of the way you build neuron together. But we're going to talk about this in the afternoon, so I'm just going to explain you what the individual motif consists of. So feed-forward excitation is probably the easiest motif that you can imagine. It's about hooking excitatory neurons at each other so that when you stimulate the first neuron, it is able to transmit information to the second neuron, that is able to transmit information to the third neuron. So we can think of this as a way in which neurons are able to propagate information in sort of a straight line throughout the brain. And this is maybe the building block of how information is transmitted to the brain. This is really the way in which neurons talk to each other and single area talk to each other. For example, the neurons, the sensory neurons that are in the retina are connected to the optic nerve through the thalamic nuclei, which in turn are hooked to, again, a feed-forward excitatory input to V1, that then will split into a dorsal stream and a ventral stream that right now shouldn't tell you much, but you will know what I mean by that tomorrow. And this is something that really goes and moves along into the cortical hierarchy. This is a very famous schema that was drawn in the 90s that tells you how information that is encoded at the level of the retina from the retinal ganglion cells moves up to the primary sensory area, to the secondary sensory area, tertiary, fourth, and then all these association area to end up through the internal cortex into the hippocampal formation. All these connections that are drawn here are mostly excitatory and based on principle of feed-forward excitation. With feedback excitation, instead, we define a motif in which a neuron connects to another neuron, which by this perspective can be considered to be postsynaptic to this presynaptic neuron. But this neuron at this moment creates a branch of its axon that goes back and makes a synapse onto the neuron that receives information on. This is actually a really important motif because I talked yesterday in my group. This is a way in which you can create information that persists long after the stimulus is applied to the first neuron. For example, if you give a stimulus here, you excite neuron number 1, that is the input neuron of this circuit. This input neuron excites neuron number 2, which is the output neuron of the circuit. At this moment, you don't give the stimulus anymore. Neuron number 1 should be silent, right? Because it's not stimulated. But the fact that it receives a synapse from the postsynaptic neuron makes sure that the information is relayed back, so it's excited again, and so on and so on and so on, that makes sure that there is a persistent activity into this network long after the stimulus that has elicited that activity is actually stopping to exist. And you have an example of this into your working memory. You look at some pattern of, I don't know, a series of numbers, or someone tells you to hold on the phone numbers in your mind, and you continually and persistently keep this information in the workspace of your prefrontal cortex in order to basically be able to recall it at a certain point. In this case, you see a task in which a mouse is given a stimulus at this time, in which there is a delay, and in which it has to activate a response. For example, I don't know, the stimulus might be press the level of on your right or on your left. You see that the neuron is able to be specifically keeping the information press the level on your right, which is encoded in this blue trace here, until the response is released. And it's extremely specific also to the type of message that is relayed. So by having this persistent activity in area of the prefrontal area, the animal is able to solve the task of keeping information in mind in order to get a reward, that in this case could be sugar water. Are we clear on recurrent excitation or feedback excitation? All right. The third is a combination of two different motifs, and this is convergence and divergence. This is a little bit more complicated than feed-forward excitation. In feed-forward excitation, I showed you that there is one neuron connected to another neuron and so on. And you can think of these about, for example, neurons that connect across area, right? The dentate gyrus connected to CA3, the CA3 connecting to CA1, the CA1 connected to the internal cortex. In this case, you have either that several neurons are able to connect all to the same postsynaptic neuron, or that one neuron is able to create different branches to transmit its information to different neurons. And these branches are usually called collaterals, right? Convergent and divergent connection allow individual neurons to integrate input from multiple presynaptic input or to broadcast one information to multiple neurons. And one concrete example of that is having coincident detection. We said yesterday that the activation of one synapse is not sufficient to be able to drive the activity of a postsynaptic neuron. In this case, if you have a convergent stimuli with one neuron that tells you, look, there is an alternation of black and white, and another neuron that tells, look, there is a horse, the postsynaptic neuron, you say, oh, maybe it's a zebra. So if you have neurons that are tuned to different features, via convergent excitation, they're able to combine these different features in order to create a response that will be specific just to the combination of these two features, but does not respond to the two features in isolation. At the same time, when you have an alternation of black and white, it can be the colors of a zebra, it can be the colors of a zebra crossing, it can be the colors of your favorite sport team when you go to the stadium, right? That neuron might transmit information to different postsynaptic partners in order to create neurons that respond to specific combination of information. Of course, again, this is an oversimplification of the system, but it makes the point that you get integration of information through convergence, and you might think of it as sort of a generalization of certain information through divergence.
One question.
Yeah.
What happens in the brain when you think about it for a bit longer, and then you get more animals with similar shapes? So, for example, the Dapi also has these tribes and other animals, but when I talk, the zebra was the first thing that popped up, and after 10 to 15 seconds there were more animals. What happens in the brain?
Oh, that's a fantastic question that we're actually really interested in the lab. So what you do is that at the beginning, whenever, it's a little bit more complex, and it touches themes that we're actually going to touch upon tomorrow about the way hippocampus and prefrontal cortex work. But you must think that you start from individual experiences, right? You see for the first time a zebra, then you see for a second time and something like that, like the animal that you were saying that I didn't capture the name. You see the Juventus logo, which is not my favorite theme for whoever is wondering that, but let's admit it is. And you have this individual experience, right? At a certain point, you start to extract from this individual experience some larger category that is having something that is an alternation of black and white, through a phenomenon that is called, the memory that you create is called semantic memory, which is basically like a memory of a category of things, not just one specific things, through a process that you might call generalization. So in the end, you have different representation of the same stimulus in different brain areas, some of which are more tuned to the specific animal and to the specific instance of you seeing that specific animal. And some of those are more general and get activated when something that belongs to the same category is actually present in front of your eyes. This is a process that is extremely fascinating to think about, because it's something that allows us to basically either transfer knowledge, but I would say between one specific category, to lump stuff that belong to the same category together. And it is the one thing that it comes back to one of the few last slides on Friday that might still allow us to consider ourselves smarter than the machine we built. Did I explain answer your question in a satisfactory way for now? It's going to be more thorough tomorrow. You're going to see how we're going to create representation that are more and more detached by the specific sensory stimulus as high we go in the cortical hierarchy, up to the point that in a specific area of the brain, you will create a representation for the concept of a zebra and not specific to zebra itself. Okay.
I have another question.
Yeah.
About the convergent motif.
Yeah.
Do all presynaptic vesicles have to be active, or can it also be that only a subgroup is active, and then how would the postsynaptic neuron differentiate between these subgroups?
Yes, so if I understand your question correctly, is if you have, for example, in the motif before, do all the three neurons need to be active to activate neuron number one, right? Okay, so it is possible that also only neuron number, these two are sufficient to activate this neuron here. You don't need to have the third neuron active, okay? That's definitely possible. How it differentiates when 1 and 2 are active versus 1 and 3 are active is because if this is the next lecture. So this is through the phenomenon of potentiation of synapses. Because 1 and 2 have been repeatedly co-active, they have really made the synapses between each other and with neuron number 3, which is this neuron here, the output neuron, stronger and stronger. So the activation of these synapses in the end will induce a depolarization in the postsynaptic neuron that is higher than the threshold and induce the action potential. The activation of 1 and 3 would not be sufficient to spike the output neuron. And this is something that you actually will do today in your exercise. But it's a phenomenon of plasticity, right? It depends on experience, it depends on the pattern of which these neurons are basically active. But there is something funny going on. That is the next motif. So I'm just gonna go and take the chance of your question, for introducing the other motif, that is the recurrent excitation. So in the recurrent excitation model, also considered lateral excitation, you have two layers of neurons. Some are input neurons, and familiarize with this terminology because it's something that you're going to be seeing from now on until Friday a lot. So you have two neurons that are here, and you have another layer of neurons that are basically the postsynaptic partners of these neurons. Now, the postsynaptic partners are able to cross and excite each other, so that when one of these neurons is excited, for example, either neuron A is excited, these two neurons are active, or whether neuron B is excited, these two neurons are active. Why is that important? Because of the pattern of the way in which these neurons are connected and who excites who, if you have tons of this type of lateral excited neurons in each other, you can create and stabilize one pattern of activity and use that to store some information. For example, let's go back to our favorite circuit, the most beautiful circuit in the brain, which is the entorhinal hippocampal network, and to the CA3 area that we have talked before. We said before that one CA3 area, it's able to listen to 12,000 different others. One neuron in the CA3 area is able to listen to 12,000 other neurons in the same area, right? So in the end, you create a very large matrix of connection that you can see here, in which the crossing of each axon with the dendrite is a synapse. Now imagine that you are in the Münsterplatz in Basel, and you see the Münster, right? And you activate a certain pattern of these synapses so that, for example, neuron number 1, 2, and 3 are active at the same time, but neuron number 4 is not. Those synapses, because of repeated activation, and we'll see in the next lecture why and how, they become potentiated so that they create a specific pattern of activity into the CA3 area of the network, so much so that the activation of each one of these synapses will be sufficient not only to activate the other synapses, but also to activate and retrieve the whole combination of neurons that was active when you saw the Münster. So in the end, even if you give just a small detail of the Münster, which is the one that activates neuron number 3 here, because of this recurrent connectivity, this neuron number 3 will be able to bring online the whole neuronal ensemble, and this is again something that you will see in the next slide, in the next lecture, and induce you to retrieve and recall the whole image and say, oh yeah, this is the door of the Münster in Basel. Are you agree with that? So basically, you need to have the activity of the whole group of cells during the acquisition phase, but during the recall phase, you can have the activation of a subset of that group and still be able to complete the information that is missing because those synapses have been made stronger to begin with. This is one of the ways in which our memory works, and we will talk about this more on Thursday. Okay. All right, now we switch gear, and so far we have only talked about motifs in which excitatory neurons are connected with each other. Now we throw in a little bit of inhibition, and we see something interesting happening. That is, well, we think, and I think we are correct in thinking that the way in which we connect excitatory neuron decides how information is propagated, how information is stored and recalled. When we think about the inhibitory neuron thrown into a network, we think about how we change the way in which neurons spike. So in the end, the inhibitory neuron are the ones that have a higher weight in processing information into our brain, while excitatory neuron have a high weight in transmitting this information from area to area. But this is just a conjecture and hypothesis that our lab and many other labs in the world are testing at the moment. Anyway, we have the same type of terminology as before, feed-forward and feedback, but this time applied to inhibition. Feed-forward inhibition is a connectivity motif in which you have an excitatory neuron that activates a postsynaptic neuron, and at the same time activates an interneuron that inhibits the postsynaptic neuron. Feedback inhibition instead is a motif in which an excitatory neuron activates an inhibitor neuron that inhibits itself. In the brain, we find many times this type of connectivity motifs because they control the duration of the magnitude of the excitation of the postsynaptic neuron. What I mean is that they put a temporal limit to which the postsynaptic neuron can respond. For example, imagine that this neuron is constantly active and is constantly activated this neuron. The neuron that is constantly active here induces an activation of the postsynaptic neuron, so this also will start to be active. But this activity will be cut in time because at a certain point, with a delay of 1 synapse, you have an inhibitor information coming down that will be factored into the postsynaptic neuron to temporarily limit its activity. This is something that you will look in the afternoon through your exercises. But one thing that I want to bring forward, because we talked about it yesterday when we talked about oscillation, if you were not completely in delta waves, that is the sleep wave, you know that there is this oscillation of activity. The way in which oscillation are created locally in many brain areas is through an interplay between excitation and inhibition. Conceptually, but this is not the way in which the oscillation is actually created, but conceptually, this oscillation has been modeled as having a source of excitatory activity activating a source of inhibitory activity that feedbacks onto the excitatory activity itself. So imagine what happens. This excitatory source becomes more and more and more active. When the activity increases, this increases also the inhibition that is exerting onto the excitatory neuron. So excitatory neuron goes up, but now at the peak, inhibition also goes up, so excitatory neuron goes down. But having minimal excitatory activity means that there is minimal inhibitory activity. So the excitatory activity can also increase, and so on and so forth, by creating the oscillation. You should have seen this type of principle of having a source of excitation inhibition that interact with each other in many realms of biology. The circadian cycle is one way in which this happens. Excitatory, like an answer and repression of genes into a cyclic way is also a way in which this happens. This is a pervasive type of connectivity motif that does not apply only to the brain, but applies everywhere in biology. This in the brain is particularly important because it creates those oscillations that we saw yesterday, that we're not going to see anymore. All right, we move to another recurrent inhibitory motif. This is recurrent inhibition. And this is what I was already explaining you before. You have two different streams of excitatory information. You have two different streams of feed-forward excitation. Oops. And you have in the middle a hidden layer between the excitatory inhibitor layer that is able to cross-inhibit the other pathway. So that when pathway 1 is active, pathway 2 is shut down, and when pathway 2 is active, pathway 1 is shut down. Can you imagine where this can be used?
Sympathicus and parasympathicus.
What? Sorry, Fabio, I didn't hear you.
In sympathicus or parasympathicus, so for example, if you have stress, the sympathicus is shut down, and the other way around.
Absolutely, absolutely. I was thinking more about something that was within the nervous system, like activity of neurons, but this is definitely one, Fabio. Like, if you need to have two things that are mutually exclusive with each other, this is the way that you want to do it, right? You either produce one or elicit one response, and that shut down the other response. In fact, it's so pervasive that it's also possible that this type of motif is used by two opposite memories to actually inhibit each other, so that you activate one at the same time use that particular memory to inhibit the other one. A more, let's say, immediate or simple is your movement. To move something like your arm, your leg, and something, you have to coordinate the ability to contract a flexor movement and release and basically relax an extensor movement muscles. And this is the way in which basically this is activated. When you have the excitatory information going through the flexor motor neuron, you inhibit the extensor motor neuron. And when the extensor motor neuron is active, you inhibit the flexor motor neuron. So you see this alternation in activity that make you walk, move your arm, or do whatever you want to do at the gym now that you can go from yesterday, right? So this is a mutual exclusive pathway of information. You employ this motif. One of the last one is lateral inhibition. Lateral inhibition, as the name says, you have an input layer, you have an output layer, and then you have an interneuron here, which is excited by the input layer and is able to inhibit the output layer, right? Which is also extremely important because it's one of the most important and deployed connectivity motif in our sensory organs. For example, in the retina. Lateral inhibition is really used to increase the sharpness and to allow you to detect sharp boundaries between the stimuli. For example, edge detection is the way in which you define that there is an edge between a bar that is darker and a bar that is lighter. This is one of the ways in which your visual system increases the contrast between stimuli, but is also a way in which you are basically increasing contrast between different types of stimuli which are adjacent to each other, so in which you need to create a separation between each other. This particular type of configuration gives rise to a very nice optic illusion because the retinal cells in the eye that receive the stimulation from, for example, like a greater stimulation are able to inhibit the cells to a higher degree than cells that receive less intense stimulation. So if on the two sides of an edge, you have different level of activity and means different level of inhibition, you might have mismatch between these two parts of the circuit that creates the impression of seeing an edge of a different color than what it actually is, or to not be able to define the specific color of the transition phase. In this case, for example, look at these stripes and then look at this line here. What the bug that I am telling you now makes you think is that this gray line here is not of an even color, but is created by the alternation of a lighter area or of a darker area, of a lighter area, of a darker area, and so on and so forth. When in reality, if I was able to isolate the surrounding activity so that you basically don't have this unbalance between the inhibition that you get from one side of the edge versus the other side of the edge, you would appreciate that this is actually of the same color. The other part is that this type of, let's say, illusion creates is make you think that the area close by to the darker area is actually darker when it is not. It should be an homogeneous color. Not in this case, but in other type of association. Lateral inhibition is pervasive in our sensory organs. It's present in the retina, but it's also present like in auditory circuit and so on. It's really like extremely important for being able to parse the stimuli and increase context and identify edges between stimuli. All right, we move to the last of our network motifs, which is disinhibition. It's quite particular because disinhibition is quite uniquely involving inhibitor neurons, although of course it's not the case. But this is a situation in which inhibitor neurons and inhibitor neurons synapse onto another inhibitor neurons, therefore reducing the inhibition that this interneuron exerts on another interneuron. Okay? To explain this, I just want to bring upon a study that has been done actually here in Basel a few years ago, or few studies actually, in which you understand how disinhibition works. Disinhibition, you usually have a pyramidal neuron and two interneurons like somatostatin and parvalbumin neuron, which are able to inhibit both the dendrite and the soma of this neuron. As a result of this inhibition, you need a very strong current to excite the excitatory neuron, and you get output in few of these cases, just when the membrane of the neuron is able to cross the threshold potential, right? So the membrane is hyperpolarized, meaning that the neuron is less excitable. There is another class of interneurons that is positive to the vasointestinal peptide, which is a class of neurons that have prevalently contact with other interneurons. This is the disinhibitor neuron. This is the neuron that synapses onto somatostatin neuron and parvalbumin expressing interneuron. A stimulus that activates this interneuron will be able to turn on and shut down these two inhibitor neurons here, and as a consequence, produce disinhibition onto the excitatory neuron, which will make the membrane being a little bit more positive, so a little bit closer to the threshold, so more excitable. And some studies in the past decades have identified disinhibition as one of the connectivity motifs that allows our brain to actually write information when you get sensory stimuli from the outside, and to increase the plasticity of neural circuits because it increases the source of excitability of some of the neurons that are present in our cortex. If you're interested in this type of circuit, let me know. We work on this in the lab, but we also, like, I can suggest you work from other labs that have investigated this phenomenon and single out the contribution of individual neurons and interneurons to this phenomenon. All right, this is what I want to say about the circuit motif. The other thing before we break for a break is that, of course, you don't have to think that these network motif are actually isolated, that in, for example, in the hippocampus, you only find one network motif more the other. The complication comes that in real biological networks, these motifs are combined to each other to create complex network architectures that we can define as network topology, which is basically like a reference to the way in which the neurons are connected to each other, which make activity or the possibility, which make possible changing the temporal dynamic of the activity of individual neurons. For example, if we look at the pyramidal cells in CA3, which again is our model, is our favorite model, you can see that there is a lot of different connectivity between positive neurons and inhibitor neurons. We deploy feedback inhibition, feed-forward inhibition, feed-forward excitation, lateral inhibition, and so on and so forth. So I'm not going to explain in detail the detail of this network here because it's beyond the scope of this week, but just to tell you that almost everything that you find here, you can find in this specific connectivity network architecture that is present in CA3. Why do we employ so many different connectivity motifs and create network architecture that are so different? We do that because connectivity motifs are able to change the temporal activity of a neuron into a predictable manner. For example, I showed you yesterday that the neuron is slightly active here, and then it gets active when a stimulus is applied, and then connectivity goes back to baseline. You can imagine that connectivity is given because a feed-forward excitatory input has arrived at this moment, and here, even though the feed-forward excitatory input might still be present, a feed-forward inhibitory input or a feedback inhibitor input has caused a decrease of the activity of this neuron in time, right? So in last instance, our connectivity motif are able to increase a neuron's activity over time, decrease the neuron activity over time, change the timing of this neuron activity in relation to the stimulus, to the oscillation. Again, we are not going to go through this, or synchronize activity between many neurons on the network to create those ensembles that I told you before, and then I will tell you in the next lecture, okay? So in the end, the ability of neurons to change their activity rate over time is what allows our brain to encode, process, and store information. And this is what we are going to talk about tomorrow when we're going to talk about the neural code. So how should you think in a very intuitive way to connectivity motifs? As a simple operation, right? A connectivity motif and the implementation can take some sort of inputs and transform this input into an output. So it's like when you tell to do a summation of two inputs, when you do a division, when you say that you do a subtraction, and so on. This is literally sometimes what connectivity motifs do, are able to integrate inputs from different sources, are able to subtract, to increase the contrast, and so on and so forth. In last instance, our neuron, we want to be able to induce increase of activity in response to a stimulus, like this neuron here, decrease of activity in response to a stimulus, no change, increase or change in the timing of the activity in relation to the single oscillation, or synchronize the neurons to speak only when the other neurons of the network speak. And we're going to see how this is important not only to store information and to make plastic connection, but it's important to create information in the brain that is basically like very far away and removed from our sensory periphery. All right, this was the last piece of knowledge that I wanted to transmit to you with this lecture. Connectivity motifs are going to be the subject that you're going to be experimenting on this afternoon. You're going to be building this connectivity motif, describing, see how they change the pattern of activity of individual neurons. Any question about this part? Okay, so if there is no question, I guess that we can move. I forgot to put the Slido up actually, so I will do it now. But if you have a question that you want to ask anonymously, you can do it to the next lecture, and I will answer to those about this lecture as well. Okay, so I think we can see each other again at 11 here. You have time to stretch your legs and drink another coffee.